Thank you so much, and thank you for that warm welcome, that great introduction. Thank you for coming out on a beautiful Friday afternoon, and thank you for the Apple box giving me the height lift that I so badly need. I really do appreciate that. What I want to do today, first of all, I'm thrilled to be here for so many reasons. But there's a special reason I'm really thrilled to be here, and that the people who have gotten this medal in the past, and I'll talk a little bit about it, have been some of my most important heroes in biomedical science, not only because of the role as scientists, but as their role as thinkers and humanists. And I'll talk a little bit about Pierre Taylor Desjardins, the Jesuit paleontologist, or Lemaitre, who got it in 1934. That's how long this medal has been around, a Belgian priest who discovered the expanding universe. And that's been meaningful to me because I've been recognized for the science, but not so much as my role as a thinker. And this is really the first time I've been recognized as someone who thinks about bigger things from time to time and reconciling science and religion, which I think is so important in our society. But I especially think it's important now because as I've been writing about for years now, we've got this increasing infiltration of health disinformation, science disinformation, and anti-science sentiments going on in this country. And we're seeing more and more prominent American scientists paraded in front of C-SPAN cameras to try to humiliate them. So we're going through this very dark time, and a number of us have been wringing our hands. How do we reconcile this? How do we walk this back? How do we bring this back? And by getting this Mendel medal, the light bulb went off, and I had lunch with your provost today, just a wonderful person. And excuse me, getting over a cold. I don't have COVID, it's getting over a cold. That I think there's a special role for Catholic universities here in this because they have the moral authority that's respected by both people on the political left and on the political right. And therefore, it could be an amazing convener for helping resolve all of these attacks against science and scientists, and just something to think about. I think this is something very special that you have at Villanova University, and you can do things here that I think few other universities can. And so think about that as my lecture proceeds. You know, what can be the role of a place like Villanova University in that dialogue? I think there's something very profound there, although I don't quite have my finger on it. It's sort of a broad stroke, but I hope you'll give it some thought. So one of the most common questions, I'm going to start here. One of the most common questions that I've been asked over the last few years is, in going on the cable news channels on CNN and MSNBC and others every night, goes something like this. The question, more often than not, is, "Hey doc, what the heck is going on?" That's literally the question. And then, okay, what do you really mean by that? And then you start to sort of tease it out, and you start to get them talking a bit. And what they really mean is, why, among other things, are we seeing all these horrific pandemic threats now? And why are we seeing this regular cadence of pandemic threats? You had SARS in 2002, the original severe acute respiratory syndrome that came out of southern China, affected Toronto, Canada. And then you had H1N1 pandemic flu in 2009, and then Middle Eastern respiratory syndrome in 2012. Then you had Ebola out of West Africa in 2014. We even had cases in Dallas, Texas. Then you had Zika in 2016. And then Ebola again affected, horrific epidemic affected the Eastern Democratic Republic of Congo in 2019, and then, of course, the biggest one of all, COVID-19, otherwise known as SARS-2. And now we've got this thing called H5N1 that started in dairy farms in Texas, where I'm from, and now many of us are thinking another major coronavirus, SARS-3, is brewing. And that's not the only thing that's happening. What's also going on, which is kind of below the radar screen, is we're seeing now a big sharp rise in vector-borne virus infections like dengue and chikungunya and Zika, particularly in Texas on the Gulf Coast. That little map at the bottom there is a predictive map of where the Aedes aegypti mosquito is going to be rising precipitously that transmits all those viruses. So Texas, all along the Gulf Coast, in Florida. We now have malaria in South Texas and South Florida again. We have leishmaniasis, we have hookworm transmission in Alabama and in other parts of the southern United States. So the question is, so what the heck is going on? And I don't have the answer, but there's a few trends that I think that we have to consider. And again, this is why Villanova is a very special place, because it forces us to have a dialogue with people outside the pure aspects of biomedicine, because there's other 21st century forces going on. And I've written about this in a book that I wrote in 2021 called Preventing the Next Pandemic, that looks at what the heck is going on. And it includes climate change as a big factor. Now everybody's talking about climate change, but what I find is not climate change, and I'll go into this a little bit more in a minute, not climate change acting in isolation, but in concert with other 21st century forces. Urbanization, it turns out, is a big one, because a lot of these mosquitoes and everything else are urban-dwelling mosquitoes. Poverty is still a big factor for reasons that we can talk about. Then at the end, I'll talk about political instability and rising anti-science sentiment. So let's look at a couple of those forces. So what's going on with climate change? So the UN Secretary General doesn't even bother calling it global warming anymore. Now he calls it we're in an era of global boiling. And if you've been in Texas for the last few months like I have, and you really get it. I mean, it's been over 90 degrees every day this October where we are in Texas. But Michael Mann, who's a close colleague and friend from the University of Pennsylvania, on the left there, coined the term the hockey stick. So if you lay a hockey stick down with the blade facing up, over the last 100 years, we've seen this extraordinary rise in regular temperatures. And you might say, well, okay, I get the climate change. What does it have to do with pandemics? Well, a few things. One, of course, the mosquitoes and the other vectors love the warming temperatures. Viruses develop faster in mosquitoes. But the other big piece of this are bats. So bats are the origin of coronaviruses. So coronaviruses are natural viruses of bats. Ebola virus and other filoviruses are natural viruses of bats. Same with Nipah virus. And what's happening with those changes in temperature, altered rainfall patterns, the bats are now moving into new habitats that are coming into closer contact with humans and intermediate animal hosts. So there's unprecedented levels of bat migrations. This is studied by my friends and colleagues at the EcoHealth Alliance. So this is one of the big factors, but the other big one that people don't talk about as much is urbanization. So for the first time now, more people live in urban environments than rural environments. That shift happened about a decade ago. And with that, we're now expecting to see the emergence of what we call megacities. These are cities of more than 10 million people. And historically, for the last 100 years or so, the megacities have been New York, London, and Tokyo. Now that's all shifting to megacities in low and middle income countries. So Mumbai, Calcutta, Dhaka in Bangladesh, Kinshasa in the Democratic Republic of Congo, Lagos in Nigeria, Lima in Peru or Bogota. So all of the growth is happening in big urban areas in the global south and low and middle income countries. So that's a picture I have in the upper left there of Sao Paulo, these massive enterprises. So with that, we're seeing these expansion in clusters of human populations. So not only are the bats coming closer to the people, but the people are coming closer to the bats as these big urban areas begin expanding and outstripping their urban infrastructure. So those are two of the really important drivers now that are part of the answer to this what the heck is going on. And again, it's going to be a place like Villanova University that has people who think about climate change and sociology and urban politics that are really going to help us solve this. And speaking of where this is happening at an accelerated pace, right where I am in Texas, where if you look at the cities of the Texas Triangle, meaning Dallas, Fort Worth at the top of the triangle, Houston in the lower right-hand corner, and Austin and San Antonio on the left-hand corner of the triangle, this is where it's happening first and foremost, this explosive growth in urban areas, but also with that heat and humidity. And that's the reason why the School of Tropical Medicine that I launched 14 years ago is based there, because we actually had all of these diseases at one time. We've been able to keep them under control, but now they're all roaring back. So now we have malaria back in South Texas. It was widespread in the 1800s in the US. It's now back. Yellow fever epidemics in Huntsville, Texas, and Galveston. Now I've written in the New England Journal that that's transmitted by the Aedes aegypti mosquito. There's a likelihood that can come back in addition to dengue and Zika and chikungunya virus. Now we have hookworm again in the South. And this is going to be our new normal for a while, this regular cadence of horrible respiratory virus pandemics as well as tropical diseases, particularly in the southern parts of the US. And so we're very much concerned about that. We're doing a lot of outbreak investigation, looking at some climate health warning systems, but we're also trying to make vaccines for this. And this is one of the reasons, another reason I came to Texas 14 years ago. This is a picture of our Texas Medical Center. Has anyone ever seen it, by the way? Just a couple of you. It's, we do everything big in Texas. This is, talk about mega cities. This is the first medical city. It's extraordinary. It's a medical city of 120,000 employees, 60 institutions, including the MD Anderson Cancer Center and Texas Children's Hospital and Baylor College of Medicine. It's the largest life science complex in the world. And I invite you all to come and see it, and I'll give you a tour of the labs. Just say you're from Villanova, and that's like the key. That's going to be as good as the key to the city, to come and see where we are. And what we did was to create not only a School of Tropical Medicine, but this very unique vaccine center to make the vaccines that the pharmaceutical companies won't make. And it's not that the pharmaceutical companies are bad guys. They provide a lot of important vaccines that are used globally to reduce the incidence of childhood infections like measles and whooping cough and Hib meningitis and others. But what the pharma companies haven't been willing to do is to make vaccines that only occur for diseases that only occur among the world's poor. And that was the ambition that we set out to do, to create this Texas Children's Hospital CVD, Center for Vaccine Development. So we're about 20+ scientists, led by myself and my science partner for the last 25 years, Dr. Mary Elena Bottazzi, to make vaccines that ordinarily the pharma companies won't touch because there's no obvious financial return. So we began making vaccines for hookworm anemia that I started when I was your age, as the student's age, as an MD PhD student in New York 40 years ago, for schistosomiasis, for malaria and Chagas disease. And then we got approached by a group around 12 years ago from the New York Blood Center. Shibu Jiang and Lan Yingdu came to us and said, "Hey, Peter, just like nobody cares about your parasitic disease vaccines, no one cares about our coronavirus vaccines, meaning vaccines for SARS and MERS, the original ones in the 2000s. Will you work with us?" And so how could I say no, right? So what a great offer. So we wrote a grant to the NIH, it got funded. We began making SARS and MERS vaccines. And then when the COVID-19 sequence came online on bioRxiv in January 2020, we pivoted and made a low-cost vaccine for COVID that reached 100 million people. So I want to talk about that as well, because it provided a way to say you don't only have to be a big pharma company to do big things in the vaccine space. There are other ways to work through universities and academic health centers. So I'll tell that story. We sometimes call our vaccines the anti-poverty vaccines, and that's because trying to find ways to get people, to the policymakers, to care about our diseases. Many of them are chronic and debilitating conditions. They cause impaired worker productivity, bad adverse pregnancy outcomes. They disproportionately affect pregnant women. They cause children to not grow and develop normally as they should. And so this was the first book I also started writing book. This is the first book I wrote about this idea. It was called Forgotten People, Forgotten Diseases, which my adult kids used to call Dad's Forgotten Book on Forgotten People with Forgotten Diseases, although now it's gone into its third edition, so the joke's on them. Don't be careful what you say to your parents. And with that, some interesting spin-offs. So, I was very inspired by, being honored by Villanova University. And I'd been writing a couple of papers looking at where these conditions, which we label neglected tropical diseases, occur. And it turns out they disproportionately occur in Catholic majority countries, which is something that's not intuitively obvious, but places like the Democratic Republic of Congo, Angola, the Philippines, these are all Catholic majority countries. And again, I think this is going to be a very important role for Catholic universities to take on these parasitic and neglected tropical diseases. The Bill and Melinda Gates Foundation estimates that by the year 2050, 40% of the world's poorest people will live in two countries. Will live in Nigeria and the Democratic Republic of Congo. DR Congo, of course, is a Catholic majority country. Nigeria's got a huge Catholic population. Certainly something, I think, to give some thought to. And of course, very inspired by one of my heroes, Taylor Desjardins, who was very passionate about writing about science in the bigger space and what science meant to humanity. He was a paleontologist from France, educated at Sorbonne, and worked at the Catholic, what was then called the Catholic Institute in Paris. I think it's called the Catholic University now. Worked all over China and Asia. And so I wrote actually a paper about being inspired by Pierre Taylor Desjardins. And then I tried to get it published. Boy, that was hard because I'm not a social scientist, so it doesn't really, I don't really speak the language of social science, so I couldn't get it into a social science journal. And then a typical biomedical journal would look at this and say, what is this? What's this guy, what's Hotez writing about, Pierre Taylor Desjardins? Finally, the Linacre Quarterly, which is a Catholic medical journal, agreed to publish. I was thrilled to finally get it there. So an inspirational figure. So getting the medal, the same one that Pierre Taylor Desjardins got in 1937, I was just over the moon when you invited me to come here and do this. Now, what we try to do is we do the cool and innovative stuff like mRNA and particle vaccines, but we also do something different. We try to be compatible with vaccine producers in low and middle income countries. And they're represented by the blue dots in Asia, on the African continent, and Latin America. And they actually band together, they call themselves the Developing Country Vaccine Manufacturers Network. They meet annually. You can go to their website, dcvmn.org, and they just met in Sao Paulo a couple of weeks ago. And it turns out one of the most common technologies that they make is they all make their own recombinant protein hepatitis B vaccine that most of you got as kids. It's actually done through a process of microbial fermentation in yeast. What you do is you take a gene and you put it in either into baker's yeast, Saccharomyces cerevisiae, or a methanol-utilizing yeast like Pichia or Insenula, and then you grow it up in a big vat, in a big bioreactor. If you ever visit a brewery, it looks very much like the vaccine producers that make hepatitis B vaccine, because it's the same fermentation process. It's a well-tested technology. It's one of the cheapest technologies out there, low cost. The vaccine antigens they produce usually just require simple refrigeration. You don't need any onerous freezer chain. Parents trust it because they've been vaccinating their kids with hepatitis B vaccine. So it checks a lot of boxes if you want to be in the business, if you want to call it that, for global health vaccine. So Mary Elena and I came up with this idea that if we're going to be making low-cost vaccines for the world, yes, we'll try to do mRNA and particle technologies, but where we can, we want to see if we can make this through microbial fermentation in yeast to make recombinant protein vaccines. Another advantage is it's a vegan process. No animal cells, human cells, animal protein, human cells, which is also turns out to be very helpful. The only problem with it, not the only problem, but one of the problems with it is, like all scientists, including scientists here, we apply for NIH grants, National Institutes of Health. It has to go through study section. And the problem is we often will get dinged by study section because they'll say, hey, Peter, great humanitarian advance, but what you're proposing to do is not very innovative. And they're right. It's not because it's an older process, microbial fermentation in yeast, but that's what you need to do to make a low-cost vaccine for the world. So these are the kinds of struggles that we have doing this as a nonprofit in an academic health center, that sometimes the NIH is not, even though they think it's a great idea, it doesn't check the innovation box because it uses an older technology. But what we do is we'll make the production cell bank and literally send it via world courier to the vaccine producer in a low and middle income country, more often than not in Asia, and now more recently on the African continent. And then we'll do the scale up at around the 5, 10, 20 liter fermentation scale, and they'll do it at the 1,000 liter fermentation scale, and we'll get on a Zoom call with them to work with them on it. And you know what that means. If you want to get to them by close of business their time, it's a lot of 4 or 5 a.m. Zoom calls, but that seems to be a fairly effective way to do it. And so these are some of the signature diseases that we're working on. Hookworm infection, schistosomiasis, and malaria. And I'm showing you, I don't want to have this go into a microbiology parasitology lecture, so I won't go into the details of how these diseases are transmitted, but I'm doing it to show you the maps of where they occur. So on the left, hookworm and schistosomiasis in the purple there, on the African continent, Southeast Asia, and Latin America. On the right, where hookworm and malaria occur, co-endemic on the African continent. And now take a step back and imagine if you're the chief executive officer of a biotech or a big pharma company, right? This is probably not the map you want in your business plan, right? Because where's your financial return? Your financial return is going to be in North American countries, in the US and Canada, or Western Europe or Japan, not where the sweet spot where our diseases occur. So this is one of the big challenges we face, not only doing interesting science, but what is the sustainable business model? And the answer is, we don't really know. So we're kind of doing this without really knowing where we get to at the end. And again, this is one of the great things about Villanova. You're blessed with a business school where you have a lot of smart people who can think about how to solve problems like this. So this is one of the big vaccines that we work on called human hookworm infection, hookworm anemia. That's actually a picture of a hookworm. It's not a virus. It's not even a microbe. It's actually an animal. It's a worm about 1 centimeter in length that has teeth and cutting plates that allow it to burrow deep into the intestinal lining and then extract blood out of the intestinal wall where there are blood vessels, capillaries, and arterioles. And this is now a leading cause of anemia because it causes blood loss and ingests the blood in low and middle income countries. This is a movie taken by a colleague of mine, a gastroenterologist in Hyderabad, India, who was doing endoscopy and found, not surprisingly, hookworms in his patient. And this is what a hookworm looks like. So you see the blood pouring around the outer edges of the worm's mouth. It's a sort of a messy feeder. It's tearing into capillaries in the lamina propria, and then the blood courses through the body of the worm. And that barber pole effect you see with the white, those are the white parts of the reproductive organs of the worm, and the blood is passing through the gut, and it's causing blood loss, leading to anemia. And by this mechanism, it's a leading cause of anemia on the African continent, in India, and elsewhere in South Asia and Indonesia. The other aspect of it is you become infected with hookworm because of the larval stages that are in the soil. So if you pick up a handful of dirt in Africa or in India, you'll pick up lots and lots of those hookworm larvae. They're dormant larvae. They're about 0.5 millimeter in length, so you can't really see them easily with the naked eye, but you can see them under low power microscopy. And they're very hardy. They're very thermotolerant. So one of the reasons we're seeing so much hookworm now is as the soils on the African continent are baking, or the soils in India, the hookworm larvae are like, yeah, that's fine with us, because they're so thermotolerant. So this is why it's becoming such a dominant parasite. And back when I was an MD and PhD student in New York City at Rockefeller University and Cornell, I wrote this article with my boss, Anthony Cerami, on the idea that we can make a vaccine for hookworm using recombinant protein antigens. This was when molecular biology was first starting off, and this turned out to be a 40-year journey, doing a lot of basic biochemistry on the hookworm, trying to figure out how hookworms feed on blood and then trying to interfere with it. So what I'm showing you is a hookworm attached to in the intestine, lacerating capillaries, and then the red blood cells pass through the body of the worm because the worm likes to ingest hemoglobin. So the hemoglobin inside the red cell is the nutrient source for the parasite. So what it does is, that's a cartoon of cross-section of the gut of a hookworm, which is lined with enzymes involved with hemoglobin digestion, a series of proteolytic enzymes bound by the brush border membrane, bound via GPI anchor sequence to the brush border membrane, and then it starts degrading hemoglobin in stepwise fashion with an aspartic protease, a cysteine protease, a metalloprotease. And then the animal, the hookworm, has to do something about heme, the hemoidy, the part that has the iron in it. That's what gives hemoglobin its red color. It turns out if you feed on blood, you don't like heme, because heme generates toxic oxygen radicals, it peroxidates lipid membranes, it breaks apart DNA. So anything that feeds on blood, whether it's a leech or a malaria parasite or a hookworm, has evolved mechanisms to detoxify the heme. So the malaria parasite does it from the inside. It's feeding on hemoglobin in the red blood cell. What it does is it converts it into an inert pigment known as hemozoin, or it has a specially modified enzyme called a glutathione transferase that has got a specially adopted heme binding pocket, and that's what hookworms have. And so the reason I'm telling you that is, over the years, we've tested all of these enzymes as prospective vaccine candidates, as recombinant proteins, to inject them into a laboratory animal as a vaccine and then see what happens after they become infected with hookworm. And it turns out that the GST molecule, the glutathione transferase for heme detoxification, was the best vaccine. And Toya Nasojo in our group solved the X-ray crystal structure of it and showed it's got this unique heme binding function. And it was a great vaccine in the laboratory. And then we had some initial funding from the Bill and Melinda Gates Foundation, as well as the European Union, including the Amsterdam Institute of Global Health and Development. The Dutch government was giving us support. And we were taking these enzymes from hookworms through a series of phase 1, phase 2 clinical trials. And then we got stuck. And the reason we got stuck was we needed to get to a phase 3 study, a large study, to show that it works. So if you remember, when the mRNA vaccines were being tested in the end of 2020, it was the Pfizer, Moderna vaccines were done after 40 to 60,000 person studies. Those are very expensive studies, tens of millions of dollars, but of course Operation Warp Speed of the federal government was willing to pay for it. With us, they say, well, you want us to invest tens of millions of dollars on a hookworm vaccine with no financial return. And you need to do something to de-risk this. Basically what they said to us is, Peter, we don't really want to invest in your large phase 3 trial to see if your hookworm vaccine works, ... unless we already know it works. So what the heck do you do with that, right? And so they said, we've got the answer for you. What we want you to do is do a human challenge infection model. They call it CHIM, Challenge Human Infection Model, to infect human volunteers with hookworm and show that your vaccine protects them. It should take you about 7-8 months. So, we did it. But it took us 7-8 years, unfortunately. By that time, our donors had gotten sort of tired of us, and they moved on. But Jeff Bethany and David Demert, our colleagues, worked out a human challenge model. What you do is you take 50 hookworm larvae, put them on the skin. They penetrate through the skin. They go through the afferent circulation, through the blood vessels, to the inferior vena cava, the right side of the heart, and then through the pulmonary artery, through the lungs. They break out of the lung capillaries into the alveoli, climb up the alveolar tree, cross over the trachea and epiglottis and swallowed, become adult blood-feeding hookworms. And that's when eosinophilia starts. They start inducing the production of a type of white blood cell known as an eosinophil. And then they cause blood loss. And it took them 7-8 years to work this out. This is done through the FDA. This is done through an investigational protocol from the FDA. And it's not easy because you have to have an infected volunteer where the eggs and their feces are being collected. You have to hatch the eggs, clean up the larvae, and then do them under the right circumstances to give them to human volunteers. They did this, and they said, okay, now we're ready to test a hookworm vaccine. I was a little skittish initially because if you think about it, if you're giving 50 hookworm larvae, how many of them are becoming adult worms? Probably about a dozen, and there's variation. And the problem is to get good statistics, you would need a vaccine that protects close to 100%. But we went ahead, and there was something very providential about this. It worked. These are some results looking at the quantitative egg counts because these worms may produce eggs. On the left is the control. I believe it's the blue one headed toward the right is our recombinant GST protein on alum, and the one over that's flatline at the bottom is our recombinant protein on alum together with a CpG oligonucleotide. This is a TLR9 agonist. It's got 100% protection, and moreover, it correlated with antibody responses on the right, and it stopped the eosinophilia as well. So we were like, oh my God, this is incredible. Now here's the problem. Now we've got a vaccine that we show works, and it's de-risking it, but we still need a vaccine producer to come along and scale it up and confirm the studies, and then do the distribution. And we did this with COVID, and I'll tell you about it in a minute. We did it with 2 vaccine producers, Biological E in India and Biopharma in Indonesia, and they scaled up and produced our COVID vaccine. But they did that because the Indian government and Indonesian government said, hey, if you show this vaccine, you can scale it up and show it works, we'll guarantee the purchase of 300 million doses. So the point is that sort of de-risked the investment of the companies because it was COVID. But now you do something like a hookworm anemia vaccine, we don't have that guarantee yet. So this is spending a lot of time going, I just got back from India a while ago, having those discussions, or at the World Health Organization in Geneva, or with the National Institutes of Health to figure out what is that sustainable business model. And that turns out to be one of my biggest challenges right now. But with COVID, it was a bit different. So one of the vaccines that our 2 vaccines that we developed, very similar. One is a recombinant protein vaccine in yeast, just like the hookworm vaccine. In India, it was named Corbevax. In Indonesia, it was named IndoVac. And this turned out to become the vaccine that went into the arms of 75 million adolescents as their primary immunization. And that was one of the first times, I think, that a vaccine produced in an academic health center actually went into arms on that scale. The story behind it goes something like this. This is what the world looked like in 2021, where people were not getting vaccines. So if you remember, the vaccines first rolled out at the end of 2020, Pfizer and Moderna, and by the middle of 2021, any American who wanted to get an mRNA vaccine could. They were freely and widely available, but not so much on the African continent or South Asia. This is a bubble map of the bigger the bubbles, the larger the population not being vaccinated, and it's the usual suspects on the African continent, India, Pakistan, and South Asia, Indonesia. So why was that? Well, part of the reason was Operation Warp Speed, which was well conceived, was very focused on speed and innovation, meaning they wanted a vaccine made quickly that could be distributed quickly, and they got, and they wanted to foster innovation, and they did. They got some amazing technologies, the mRNA vaccine, the particle vaccine from Novavax, the adenovirus vectored vaccine from J&J. The problem, though, is when you focus on a brand new technology, as any engineer will tell you, when you do that, there's a learning curve before you go from 0 to the 15 billion doses that were needed for the world. And what happened was predicted and predictable. Just enough doses were made to be bought up by high-income countries. It was bought up by the US government, the Canadian government, Western European countries, Japan, and that left the low and middle-income countries without any vaccine. So we were getting, and they knew we were working on a low-cost version, so we were getting frantic phone calls from ministers of health and ministers of science all over the world. Can you help us providing us with your prototype, and then we'll scale it up? And so this was what my life was like in 2020 and 2021, doing that technology transfer. And again, to do this, when you do business with Asian and African countries, you're getting up at 4 or 5 a.m. to get to them at close of business, their time. And then, at that time, I was talking on the cable news channels till late in the night, talking to Don Lemon on CNN and Lawrence O'Donnell and Brian Williams on MSNBC. So I didn't sleep for 3 years, but we were able to get this done. The other problem we face is funding it, because we were cut out of Operation Warp Speed because we weren't a pharma company. And being in Texas, we were able to get support from the King Ranch, the Clayburgh Foundation, the MD Anderson, the Dunn Foundation, the Pickhour Foundation based in New York. Tito's Vodka based in Austin, Texas gave us $2 million. Not that I'm endorsing alcohol consumption, but should some of you over the age of 21 have a vodka-containing cocktail this weekend? You don't want the Russian stuff anyway, right? So Tito's Vodka based in Austin, Texas. There you are. But that was enough. So we didn't raise the billions needed, but we were able to raise $8 or $9 million to do this technology transfer. And then when we transferred the ownership, our idea of decolonizing the vaccine ecosystem. We basically said, okay, you guys own it. We did this with no patent, no strings attached. And then he said, it's up to you to work out with your national regulatory authority in India, the FDA equivalents called the DCGI, similar in Indonesia. You work out how you're going to do your clinical trials plan, how to do your data sharing, how you're going to get approval by the World Health Organization. It was very successful. They showed that the level of virus neutralizing antibody was right up there with the mRNA vaccines and better than the adenovirus or the inactivated virus vaccines. They did step-down studies in kids. And by this time, 2 years ago, even earlier, more than 75 million adolescents had been immunized and another 20 million adults, so 100 million doses had gone out. And then we had done something similar with Indonesia. Indonesia also has vaccine production capability. They're big vaccine producers known as Bio Farma. What they did also was when they learned it was a vegan process, their ears perked up because they came into our labs and inspected all of our lab notebooks and confirmed that the every reagent used was from a non-animal, non-human source. No animal proteins, no human proteins, no animal cells, human cells. And Indonesia is a Muslim majority country, so they went back and worked with their clergy and got this certified as the first halal COVID vaccine, which something I never would have expected. When you're going to scale, things start to happen that you never would have thought. So that was very meaningful for us as well. The problem that we face now, and I'll transition over to talking about some of the anti-science, is we're not being as effective at vaccinating the world's children as we used to be. We've had 20 years of success through the Global Alliance of Vaccines and Immunization and gradually reducing the number of kids dying every year from measles and polio and diphtheria and Hib meningitis. And now we've hit a snag. And the World Health Organization is alarmed by this. We're starting to see more and more breakthrough measles epidemics, pertussis epidemics, and even polio in wastewater, even wild-type polio in Afghanistan and Pakistan. So they're becoming alarmed. So they're now doing this big push that they call the Big Catch-Up campaign. Literally, they're calling it the Big Catch-Up. The other name is the Immunization Agenda 2030. But I don't think that's the only thing going on, because the thinking was, well, it's the social disruption from COVID, the diversion of resources, supply chain issues, but there's something else going on. And that's superimposed on this is a rise in anti-vaccine activism. So that's a picture of me in the monk painting over there on the lower right. I want to finish by giving you a very hands-on view of the anti-vaccine movement. And the way I got involved in this is sort of interesting. So on the left there, I started as an MD PhD student in the Upper East Side of New York at Rockefeller and Cornell. And by the way, for those of Villanova students who are interested in MD PhD programs, they're not for everyone, but they're extraordinary opportunities, and always happy to have that career advice discussion with you. And I started making a hookworm vaccine as an MD PhD student. The point is, I'm more or less doing what I set out to do 40 years ago in the 1980s, make vaccines for the world. That was the planned part. But of course, John Lennon also says life is what happens when you're busy making other plans. The unplanned part was having 4 adult kids, including Rachel, who has autism and intellectual disabilities. And I wound up writing a book about it called Vaccines Did Not Cause Rachel's Autism, because that was the original assertion against vaccines, was that vaccines cause autism. And anyway, that wound up making me public enemy number 1 or 2 with anti-vaccine groups. Our good friend Robert F. Kennedy Jr., who likes to target me, publicly declared me on his Instagram the OG villain. And I'm so old and square, I had to look up what that meant. I had to Google it. What's OG villain? It means original gangster villain. So I do appreciate the Mendel Medal Committee for inviting the OG villain to come and talk to you today. But it gave me a front row seat to watching how this thing has evolved. And then the new book is called The Deadly Rise of Anti-Science. So what I want to do is finish up, because I do think we are seeing this enlarging anti-science ecosystem, beginning with vaccines, but now going on other aspects of biomedicine and into climate science, giving you my personal perspective on how I think the anti-vaccine movement has evolved or devolved over time. And this is a timeline of 25 years with version 1.0. I take you through version 1.0, 2.0, and 3.0. Version 1.0 were false claims that vaccines cause autism. That was a paper that was published in The Lancet, a British medical journal, in 1998. That was ultimately retracted because it was shown to be fraudulent, but the thread still exists. Then tell you how it became a political enterprise, version 2.0, and now how it's globalizing. So let me finish taking you through 1.0, 2.0, 3.0, then I'll stop and answer questions. So even with 1.0, there's a lot of nuance and variation. And you can use whatever metaphor you want. You could say whack-a-mole, meaning you knock down one of the assertion and a new one pops up, or moving the goalpost. I chose to use moving the goalpost there. So what do I mean by that? So the original assertion from that Lancet paper in 1998 from Andrew Wakefield and his colleagues at the Royal Free Hospital in London claimed that the measles, mumps, rubella vaccine, MMR vaccine, it's a live virus vaccine, the live viruses were replicating in the colon of kids, and somehow that was leading to pervasive developmental disorder. That's what they called autism at the time. And never made sense to me as the parent of a child with autism and intellectual disabilities, and it was shown not to be the case. The paper was a fraudulent paper. It was retracted by the editor of The Lancet, and also large cohort studies were supported, meaning that they would do these enormous studies showing that kids who got the MMR vaccine were no more likely to acquire autism than kids who didn't get the MMR vaccine. And similarly, kids on the autism spectrum were no more likely to have gotten the MMR vaccine than kids not on the autism spectrum. And that should have been the end of it. But this is when our good friend Robert F. Kennedy Jr. came along and wrote a second article in 2005 in that very important biomedical journal called Rolling Stone, where he claimed that, okay, it's not the MMR, but it's the thimerosal preservative that used to be in vaccine. This is where I get into the whack-a-mole, because you knock one down, another one pops up. So then the large cohort studies were funded showing kids who got vaccines with thimerosal, no more likely to get autism. Even very expensive non-human primate studies were supported, getting vaccines containing thimerosal, causing no neurodevelopmental changes, and that should have been the end of it. But then Jim Carrey and his wife at the time, Jenny McCarthy, started walking around with green T-shirts saying that we had to green our vaccines and space them further apart, because the real problem was we were overwhelming the immune system. So that was the next one, also debunked. Then it was alum vaccine. So it became this kind of exhausting exercise. Then it was claims that it was the HPV vaccine for cervical cancer that must be the problem. That was causing autoimmunity or infertility. How many people have heard that COVID vaccines cause infertility? Yeah, that's where they got it from. They copy-pasted the fake assertion from HPV and globbed it onto COVID. Or that now the new thing is chronic illness, which is so vague and untouchable, it becomes harder and harder to debunk. But that's how I got involved. This is a picture of my daughter when she was a little girl. That's me with her. And now she's a 32-year-old adult. And I wrote the book, which does a deep dive going into the science of showing that, as I've been going through with you, the evidence showing there's no link between vaccines and autism. But even that wasn't enough, because then parents would say, okay, doc, I get it. It's not vaccines, but what is it? So in other words, it wasn't enough to just say, don't blame the vaccines. Very logically, they want an alternative answer. This is where you get into the genetics of autism. So the Broad Institute at Harvard and MIT has identified more than 100 autism genes. Many of them are involved in synaptic connections, neuronal connections. Many of those genes are neuronal cytoskeleton genes. We wound up doing whole exome genomic sequencing on Rachel and my wife Ann and I with Baylor Genetics, and we found Rachel's autism gene, which is not the ones listed by Broad Institute at Harvard MIT, but very similar. It was a neuronal spectrum, a neuronal cytoskeleton gene. And now, after the book was published, the Pascal lab, Sergio Pasca at Stanford, has done these extraordinary studies using brain assembloids or organoids, creating mini brains with neurons. And if you use those neurons with autism genes, you can show aberrant migratory patterns. So you get a very complete story. We don't have it all locked down. There is an epigenetic component in some cases. How many are biology majors? Any biology majors here? You ever explain epigenetics to your roommate? It's a lot of fun, right? You start talking about microRNAs and DNA methylation, histone modification. So we still have our work cut out for us, but it's getting to a very complete story. So the point is, we had taken the wind out of the sails of a lot of anti-vaccine groups, but they found a way to regroup in a way that I wouldn't have thought of. And like so many trends, it started in California and amplified in Texas. So in California, by the 2010s, so many kids were not being vaccinated because their parents were worried about them acquiring autism, that you saw this steep rise in anti-vaccine sentiment, especially in places like Orange County in Southern California, Marin County in Northern California. And what happened was predicted and predictable. You started to see breakthrough childhood infections, beginning with a large measles outbreak in 2014, 2015. And the California legislature responded by shutting down vaccine exemptions. They basically said, okay, from now on, you want to send your kids to school, your kids have to be vaccinated. And I supported that. And it solved the measles problem, but it also created a backlash where I am in Texas. And this is when it became a political enterprise. And here's where it becomes hard to talk about, because we have to talk about something that as scientists or as healthcare providers makes us very uncomfortable. We have to talk about politics. And I say makes us uncomfortable because all of our training as physicians, scientists say you're not supposed to talk about Republicans and Democrats and liberals or conservatives or red states or blue states. But, to be honest, I haven't found a way to talk about it other than to talk about it. So I talk about it or I write about it. Not that I care about your politics, but to say we have to figure out a way to disentangle the politics from the science. So what happened was, in response to that California legislation, the Republican Tea Party in my state of Texas started funding PACs, political action committees, giving money to anti-vaccine groups to raise money for candidates running on anti-vaccine platforms, and to lobby the state legislature to make it harder and harder to vaccinate our kids, and easier for parents to opt their kids out of getting school-based vaccines. And then you had podcasters like Alex Jones, based in Austin, Texas, really pile on, and it created this whole anti-vaccine ecosystem starting in the state of Texas. To the point is, we got almost 100,000 kids not getting their vaccines in the state of Texas. And this doesn't say anything about the homeschooled kids we have in Texas. We have 700,000 homeschooled kids in our state of Texas. Anybody want to guess the percentage of homeschooled kids not getting their vaccines in the state of Texas? Throw out a percentage number for me. 10%, 80%, 100%, 95%. It's actually a trick question. We don't know. They're homeschooled, so we have no idea. But it's probably a lot, right? So we're probably well over the 100,000 mark, and that's what came off the rails during COVID-19. And here's where it gets very dark very quickly. So this is an article I wrote called "The Great Texas COVID Tragedy" that looks at the 100,000 COVID deaths in Texas. So Texas was the worst affected state. And what I'm showing you is a curve, a graph that'll look somewhat familiar to you because it probably doesn't look too different from the state of Pennsylvania. But if you look at on the x-axis there, years of the COVID pandemic, 2020, 2021, 2022, and on the y-axis are daily deaths. That first peak was our summer of 2020 when COVID hit Texas. Probably with you was closer to the spring of 2020 with the original lineage. The second peak is the alpha wave in the winter of 2021. Then the third wave is the delta wave in the last half of 2021. And then the final is the BA1 Omicron wave. Altogether 100,000 deaths. And the big green arrow sticking in the middle there is the time when COVID vaccines were freely and widely available. Anyone by the spring of 2020, probably even a little earlier, anyone who wanted to get a COVID vaccine could. And you see in Texas, almost as many Texans died after the green arrow as before the green arrow. And that's because so many Texans refused to take a COVID vaccine that was more than 90% protective against severe illness and death. So of the 100,000 deaths, close to 50,000 were unnecessary deaths. And this is why we have to care about the anti-science movement, because it's not some arcane or academic discussion. People are dying, and not small numbers, 40 to 50,000 Texans overall, and around 200,000 Americans died, mostly in states like Arkansas and Oklahoma, Louisiana, and now we know it very much along a political divide. And we can talk about the role of Fox News in promoting anti-vaccine disinformation, documented by two groups, Media Matters for America and a research group out of the Federal University of Science and Technology in Switzerland, where every night Tucker Carlson, Laura Ingraham, Sean Hannity, the most widely watched opinion news broadcasters in America, 3 million viewers a night, filled their broadcasts with anti-vaccine content. So what happened was, in their zeal to push back against vaccine mandates, which you could kind of understand, unfortunately they crossed the threshold and went the next step and falsely discredited the effectiveness and safety of vaccines. This actually began at the CPAC Conference of Conservatives in Dallas in 2021, where the rhetoric was, first they're going to vaccinate you, and then they're going to come and take away your guns and your Bibles. And as ridiculous as that sounds to us, people in my state of Texas accepted it. And what they did was they brought on all the most toxic anti-vaccine activists to falsely discredit the effectiveness and safety of vaccine. And you even had members of Congress, like Representative Marjorie Taylor Greene of Georgia, would call people like me medical brown shirts, comparing vaccines to the Holocaust, or you had Senator Ron Johnson of Wisconsin hold these phony baloney vaccine injury roundtables. So it created this whole anti-vaccine ecosystem where, if you're watching Fox News every night, or starting in the last half of 2021, Joe Rogan brought on all of the most hateful, virulent, toxic anti-vaccine activists onto his podcast on a regular basis. You're watching Rogan podcasts, watching Fox News. You went down that rabbit hole, and you made a decision based on what you were hearing that to not take a COVID vaccine, that the vaccine wasn't safe or didn't work, or ivermectin was an adequate substitute, which it clearly has no anti-COVID effects. And 200,000 Americans needlessly perished. So my pointing this out, as you can imagine, has not made me, I'm saving you all the memes that you can Google on the internet. This is the nicest one I could find. I like the comparison to Galileo, not that I deserve it, but it's better than the other stuff that you see on the internet about me. So it's taken this very dark period, and unfortunately, it's not stopping at COVID vaccines. This is playing out now in the House COVID subcommittee. that's falsely accusing scientists of making the COVID virus or leaking it from a laboratory. It's all BS. We know the natural origins on the zoonotic spillover effects of SARS-2, just like SARS. So I don't know where this is going. We're starting to see the export of this into low and middle income countries. That same US-style anti-vaccine activism is now moving into Latin America that we never saw before, moving into on the African continent. This is the new book I'm doing with Michael Mann, the climate scientist at Penn. He's been writing about the attacks on climate science and scientists. Now we're comparing notes, comparing the attacks on climate science with biomedicine. It'll be a book out next year called Science Under Siege, and maybe, when I visit Michael, come back to Villanova, we could talk about the book. So let me stop there, and happy to take questions. And again, thank you again so much for inviting me. Yes. We're trying to hand a microphone over.
At the beginning of the lecture, you mentioned how one of the
I can't see who's. Oh, there you are. Okay. Oh, hi. Hi.
Sorry.
And by the way, if you have questions but don't want to ask it in public, you can always email me. The anti-vaxxers have my email, so you can have it too. It's just Hotez, last name at bcm.edu, stands for Baylor College of Medicine. But put in the subject line you're part of this, because you can imagine what my inbox looks like these days. Yes, sir.
So at the beginning of the lecture, you mentioned how Catholic universities have this great opportunity of kind of conjoining and becoming an authority of like science and religion and being able to bridge this gap politically between the left and the right. One of the things I've seen personally, and thing I think has been observed online, is that when an authority starts to purport and like an opinion that maybe people don't support themselves, so religious people will see Catholic universities will publish, say, hey, vaccines work, let's say. They'll instead of saying, oh, they're a religious authority, we should respect them, or something like that, they'll instead say, oh, they're illegitimate, they're not real, we're gonna go to a different religious authority that will maybe be followers of Alex Jones or something like that, rather than, listen to that.
Yeah,
so
no, I get that. I mean, there's no roadmap here, right? But I do think Catholic universities are, at least as a group, and a place like Villanova is one that's not universally, but tends to be highly respected, both from groups with different political leanings, and therefore could be seen as somewhat of more of a neutral broker, where that kind of can disengage some of the politics to really talk about science. We'll see if we can get that opportunity. Yes.
Excellent talk. Thank you for being here. Two quick questions. First one, also at the beginning of your talk, you're talking about bats and urbanization had also propels kind of like increasing diseases. I'm just curious about very near you in Austin, Texas, there's a huge bat population under the bridge and a huge urban population, but I'm not sure if that's also kind of like a hot spot for something to occur. And the other one in terms of about this disinformation campaign, right? How do you also think about it? It's the limitation to getting access, right? You're talking about during this kind of like COVID campaign, like CNN seeing with things, like Fox like having other different speakers to go in there. So what's the difficulty in terms of like having the different voices going to the other medias in terms to hear a different message that the one that are typically hearing?
Yes, let me do the last part first. So, I was actually going on Fox News pretty regularly at the beginning of the pandemic, and then when the president in 2020 said that, started talking about hydroxychloroquine, how it was a miracle drug, and it was BS. It wasn't doing anything, and I couldn't go along with that. And that's when the invitations to the nighttime Fox News broadcasts ended. but the afternoon anchors hung in there with me for a while, and then Tucker Carlson and Laura Ingraham publicly started going after me, and then it became too inconvenient for Fox News. But you're right. I mean, if I would go on Newsmax, I would do, any conservative podcast or radio station or wanted to have me on, I was there, but the invitation stopped, and it all became kind of channeled to one side. The first about the diseases arising in Texas, that's right. So Texas has become a hotbed for a lot of these tropical diseases, and it's a reason why we are where we are. So thank you for that.
Thank you for a wonderful talk. I did raise my hand as a biology major here at Villanova, but that was 1981, so a long time ago. But in any event, also a practicing clinician west of here, and found myself very passionate about COVID vaccinations when we finally had them, and getting in arguments with family members, including my own mother, certain patients. And I found after a while, and as of late, having conversations with the patients, getting your flu vaccine, almost a little worn down and saying, well, this is what we know, this is what I believe as your provider. They save lives, they're going to keep you from getting sick, but I have found myself not wanting to express my passion or knowledge of science with some of these patients and my own family members. And I wonder, I mean, you must have friends, family members.
Yeah, no, absolutely.
And
so
what
it's
your talking
what
points
you're
are.
asking is very important, and this has been, I think, a big factor in the burnout of primary care providers, because it is exhausting to have that conversation. And in some cases now, patients are so militant about not getting a vaccine that they're actually firing the physician, saying, I'm not going to go see you anywhere if you keep on recommending that poison, right? Meaning the mRNA vaccine or whatever. And it's really a source of frustration. And they come, of course, downloading all this stuff on the internet, and they make you feel like you're not keeping up with the literature. You are keeping up with the literature, but you're not keeping up with the fake stuff. And it's just that the disinformation is so dominant. The way I say it is, remember, these individuals themselves are victims. They're victims of this targeted disinformation campaign. We're also learning something about the demographics of those individuals. In addition to their partisan leanings, the Kaiser Family Foundation found that one of the big factors was low educational attainment, and therefore lacking critical thinking and all those other things. So this anti-vaccine movement is a predatory movement. They are specifically targeting those individuals, if that helps any in kind of giving you empathy towards them. But it's harder and harder to do this, because in the past, back when I was practicing pediatrics, you could have a discussion with a parent and, who was pretty dug in, but have a discussion about the vaccine, why it's safe, why the diseases they prevent are killer diseases. 9 times out of 10, they'd vaccinate. Now it's all the equation, the calculus is different, because they're not vaccinating out of political ideation, out of belonging to the thing, belonging to the club, and that is really hard to break. So I think that's about the longest I don't know the answer to your question answer I think I've given in a while. But it is an enormous source of frustration. I think everyone is trying to figure out now, how do we walk this back? And that's why I thought, bringing in Catholic universities, obviously not the whole answer, but maybe that's how we begin chipping away at this problem.
Hello. Oh, hi.
Hi.
I'm actually a biology major here too. Thank you for your talk. It was very interesting. I've actually been thinking about MD, PhD programs too. And I guess my question is, what advice would you give me if I'm ever faced with a community that's not educated or low income that is anti-vaccines? Like, how would I go about this?
Yeah, I think, it's getting harder, because a lot of times they're downloading the disinformation. I think the answer partly is to get educated as to understanding what they're reading, what they're hearing about. For instance, we recently had our friend, the Florida Surgeon General, Dr. Latipo, saying mRNA vaccines are causing turbo cancers, right? And And okay, first thing you realize is there's no such thing as a turbo cancer. It's a made-up term. And then what he's saying is that the mRNA is integrating into our DNA next to oncogenes and activating oncogenes. Well, let's take a step back and say, okay, you're a biology major. The mRNA that's in lipid nanoparticles, it's getting through the cell membrane, but to get something through the nuclear membrane is tough work. That usually requires electroporation. That's why we don't have any commercial DNA vaccines. To get things through two membranes is really a challenge. Then we have proteins of our innate immune system that prevent the integration of foreign nucleic acid into our genome. Otherwise, every time we eat a hamburger, we'd be having cow DNA integrating into our genome, and so that's another piece to it. And then the likelihood of that happening next to an oncogene is even more remote. So when you go through that, you can debunk those assertions. But the problem is the disinformation is becoming more sophisticated in its nature, or in some cases becoming more outlandish. And you really have to be on top of your game and understand the science in order to debunk it. The other thing that comes up a lot, which is very frustrating, is you have a lot of physicians and even scientists bought into this stuff. And they often will go after me and they'll say, look, this is just scientific inquiry. We're asking questions. Who's this Hotez guy is being so dogmatic that's saying mRNA vaccines don't cause turbo cancers? That's not how we do science. Hotez is practicing something more akin to religion, and they even made up a term. They call it scientism, that I'm practicing scientism. So it's their use of rhetoric and things like that is becoming more clever in a very nefarious way, and so it's getting harder and harder to do this. But we need more physician scientists that can actually articulate that. And increasingly, science in medical schools is diminishing, and just because it's too crowded a curriculum. So if you really do want to become a physician scientist, an MD, PhD is a great way to go.
Hi, I'm Ruth McDermott Levy from the College of Nursing. We also need a multidisciplinary approach to this. And one of the problems is...
Especially nurses who are our most heroic frontline workers, right?
That's right.
And let's face it, people tend to trust nurses more than their physicians often.
Absolutely.
And the only one they trust more than all of us are their pharmacists. So they're also very important.
Actually, it's nursing that's been number
Is it nursing?
one.
Yeah.
So, but the other problem is the underfunding of our public health organizations and our agencies, because that puts people in the communities. And so you have that trust where you can have that conversation. Me coming in talking about mRNA and all that other stuff is just stuff to somebody making a decision for their family. So it really is not only the scientists, but the on-the-ground people to really make a difference.
And I completely agree. And I would say, the other piece to this is I think we need to do more in terms of public engagement, public communication strategies in our nursing schools, in our medical schools, in our graduate schools. Not a lot of schools do this. And I think, that has created a vacuum, and the vacuum is then filled with bad actors, filling it with a lot of disinformation. So actually providing instruction in public communication, I had to learn it by trial and error. Unfortunately, more error than trial probably, but I think that's going to be a part of this as well.
Nursing historically has done that. Medicine is finally doing that, going into the community.
Well, medicine's,
welcome.
Well, medicine's barely doing it, in part because we see the leadership of academic health centers don't even like their docs speaking out or their scientists. They tend to want to control the message, or what they'll say, where I work is better than most, but what they'll often say is, well, you're an academic, you're free to speak out, ... but don't screw this up and don't get the institution in trouble. And of course, if you're out there enough in the public domain, I promise you will screw it up and you will get the institution in trouble, and you need to know you have the institution's backing, and so we've got that whole discussion as well. There's a fantastic survey done by my colleagues at Research America, and they do this survey every couple of years, and they ask the question, can you name a living scientist? That's the question. It turns out You get the same answer every couple of years. 75% of Americans cannot name a living scientist. So most Americans cannot name a living scientist. Those who do, first they mention Einstein and Jonas Salk, not realizing they've moved on. Then when they finally get it, they'll mention Bill Nye, and Neil deGrasse Tyson. And there's nothing wrong with Bill Nye and Neil deGrasse Tyson. They do a lot of really important work, but they're not working scientists in the sense of struggling over major revisions of papers and grants and presenting findings at scientific meetings. It's a long way of saying, as scientists, we're invisible to the American people. And that makes it easier for the bad actors to portray us as sort of these lurking figures and shadows wearing white coats, plotting all these nefarious things. And we've got to somehow find a way to flip that around as well, I think.
I have one last question, or not last. Talk me off the ledge. What happens if RFK Jr. becomes the head of HHS?
Well, yeah, then I don't think it's going to happen. I don't think, first of all, he's going to get confirmed, and as HHS secretary or CDC or FDA or NIH director. But that doesn't mean he can't have an advisory role in the White House or in the Humphrey Building at HHS. Who knows what'll happen. The hope is that they'll get some good people in a CDC, NIH, and FDA director, and then that person is not necessarily going to want to be having Robert F. Kennedy Jr. looking over their shoulder, and that eventually he'd be marginalized and not have so much influence, at least on the vaccine space, but who knows. I think right now it's just too early to say how things are going to go.
Hi, what do you think is the main pushback? What drives this backlash against science and just the pushback against all of this and disinformation, and what fuels that spread?
Yeah, it's a good question. I mean, there is a financial component because some of these groups are in the health and wellness industry, and they're selling fake autism cures or nutritional supplements that no one needs. They're selling ivermectin and hydroxychloroquine after telemedicine visits, charging $1,000. So there is that financial component, I think. But why would a Fox News or a member of Congress do this? I think it's creating a form of belonging or appealing to the base. It's about political control, as far as I can tell. With attacking climate science and scientists, a fair bit of that's coming from the fossil fuel industry. I mean, as terrible as it is, at least it makes some sense, right? It's a straightforward dollars and cents. But for this one, it's a little harder to put your finger on.
And if it is against political lines, then how do you address that? How do you bridge that seemingly?
Yeah, I mean, how do you say, look, as an American citizen, you're entitled to whatever political beliefs you adhere to or aspire to. That's your right as an American citizen. But how do you say, but don't adopt this one, right? Because this one is going to cause you to engage in risky health behavior, your kids in risky health. How do you uncouple the anti-science from American politics or an American political party? And that one, I think the health sector, the scientific sector doesn't know what to do. And here's where we really need our political scientists and our smart people in government to help us figure this out. And again, it's not an academic discussion because of the loss of life that's now occurring because of it. And the fact that it's globalizing as well.
Unfortunately, we need to wrap up. Dr. Hotez, thank you so much.
Thank you again.